Okay, we're going to start with now lecture 2. This is an introduction to prokaryotic cells. This would be in chapter 3 of your textbook. One of the things you have to keep in mind is that when we look at organisms that are alive, particularly organisms that can cause disease, we have to keep in mind one basic question. Are they prokaryote or are they eukaryote? Do they have a nucleus or do they not? Prokaryotes are all unicellular, one cell. They lack a membrane-bound nucleus. They do have a chromosome, and we'll get into the details about that in a few minutes. They also lack membrane-bound organelles. For example, rough endoplasmic reticulum, smooth endoplasmic reticulum, mitochondria, Golgi apparatus, those structures. Many prokaryotes are pathogens. Now, prokaryotes include the bacteria and ARCHAEA. Now, ARCHAEA are unique in that they have some substantial structural and biochemical differences. But the key point about them is they reside in extreme environments, extreme hot water, extreme pressure, high salinity, acidic environments, alkaline environments, etc. Environments that basically you don't see now, but in some cases they did exist. On prehistoric Earth, in some cases, 1, 2, or even 3 billion years ago. And now they reside in these unique niches, hot springs, where you have volcanic oceanic vents, in huge salt ponds, or where there is a lot of unique either acidic or high pressure conditions, and we'll continue and give you further examples of that soon. Bacteria are common. They're found in many different environments. Yes, there are some bacteria that live in cooler environments, others that live in warmer environments. What I mean by cooler and warmer is relative to 37 degrees Celsius, in other words, 98.6, our body temperature. But bacteria and archaea are genetically distinct from each other. If you take a look, this is sort of a generic prokaryotic cell. As you can see in the center in purple, it has a chromosome. It's called the nucleoid region, and the chromosome is more or less just one long, incredibly long loop of DNA. Now, there will be some, from time to time, some prokaryotes that will have smaller little loops of that. They're referred to as plasmids, but the key code of DNA is in this nucleoid region. You will see from time to time other structures, ribosomes, inclusion bodies, which we will explain in a bit. But then you have to do is look at the boundary of the cell. First off, you have a cell membrane or a plasma membrane. But then to maintain the integrity of the cell, despite changes osmotically, what I mean by that is, as there's changes in solute or solvent or whatever, they have to have some type of outer structure, which might be referred to as a cell wall. Sometimes they have multiple layers of these cell wall structures. And in some cases, they will have, for some cells, a structure of glycoproteins, etc., called a capsule. Now, there are also other extensions beyond the cell membrane that many prokaryotes have. For example, some of them will have FIMBRIAE. Others will have a large flagellum, which in many cases is used to help transport or propel the prokaryote through a certain medium. When you look at prokaryotes, they actually exist in two of the basic domains of life. You have bacteria, and you have archaea. And it's believed, interestingly enough, that somewhere off from actually archaea was the development of eukaryotes. In other words, those cells, those organisms that have a membrane-bound nucleus, etc. They all seem to have had some common shared ancestor, but no one's been actually 100% sure of what. So let's get down to some of the basic details of prokaryotes. Their sizes range from 0.2, and it's supposed to be micrometer, to 750 micrometers. Remember that micrometer is basically 1/1,000,000th of a meter in length. The smallest that's been found is called a mycoplasma. The archaea has, interestingly enough, one of the largest cells in size called Thiomargarita namibiensis. It was found originally off of the coast of Namibia, Africa. There are some size limitations due to the surface area and the volume constraints. If you're not familiar with this, think about it from just this simple key point. As you increase the volume, you will have an increase also in surface area, but the surface area, well, let's say the increase is by X. When you increase by X the cell, you have an increase of X to the square of the surface area, but the volume increases X to the third. So you have really a cubed increase in whatever volume. In other words, if I say to you it's going to be 10 times larger, 5 times larger, 50 times larger. Here's the key point. You have a surface area that increases but cannot keep up with the volume of the inner cell, the volume being cytoplasm, all the enzymes, all the metabolic needs. The surface area is really the exchange site for nutrient, gas, and waste exchanges. The volume has a different demand. It demands a certain amount of gases inside, waste gases removed, glucose, amino acids, and a variety of other materials in and out. And due to the limitations of time for diffusion, for this diffusion transport within the volume of the cytoplasm, it won't get there in time. Eventually, you come to a limit in how you can increase the size of some of these cells. There is one exception to this rule, and that involves having intracellular inclusion bodies. Now, these are structures that are inside the cytoplasm, but really reduce the available volume because they occupy some of the volume and they're not doing anything in the sense of they may not be acting to have a demand for oxygen, nutrients, or produce waste. Really, what intracellular inclusion bodies are more like storage tanks. As they reduce the intracellular volume, they will occupy some of this volume with stored amount of iron atoms or phosphate or glucose, glycogen. So that's how you can get around some of that, but there's still limits to that even. Here's what I was referring to earlier. Let's say we start off with a length of one edge of a cube is 1 inch. So what do you have for surface area? Well, if you go from, let's say, 2 by 2, or excuse me, you go by 1 by 1, you end up basically, the length of one edge of the cube is 1, so you have 1 times 1 times 1. What you end up eventually having is in inches here, and I had to correct myself, I'm sorry. Let me put it to you another simple way, because 1 times 1 times 1 is still 1, and everybody goes bananas over that excuse. So let's say my length of one edge of this cube is 2 inches. So it would be 2 times 2 times 2, and that would get you upwards to a volume of 8. The surface area, which is really inches squared, would be 24. Okay, so surface area to volume is 3, 0. Basically 24 divided by 8, and you would have a factor of 3. Let's say that I want to increase it and double it from 2 on the edge to 4. So now I have 4 times 4 times 4. My surface area increases to 96. My volume increases by 64, but the surface area to volume decreases to 1.5. As you note here, what they did was they took agar cubes, okay, and they soaked them in the dye and then cross-sectioned them. And if you basically do this, you'll note that the longer it takes for the dye molecules to get through all of the volume here is the reason that you have the limit of size for prokaryotes. Hence, you will not get the 50-foot high bacteria cell that ate New York. It sounds like an old B movie from the 1950s. It takes longer for materials to pass through the volume here. As you increase in volume, you decrease you basically increase the transportation time, or you decrease the capability of nutrients passing from one area out or one area in. All right, I hope that makes it clear. I know it's a little tricky at times. Now, when we talk about prokaryotes, we've talked about size. Let's talk about shape. Now, prokaryotes have a diversity of shapes. As a matter of fact, we use some of the names as identifying factors for the particular organism. If I say something's bacilli, that's a rod or cylinder-shaped structure. If it's cocci, it's spherical shaped. If the cell is vibrio, it is comma-shaped. Stella is star-shaped. Coccobacilli is an ovoid or round barrel-shaped structure. Spirochetes are spiral-shaped, and they may move in a corkscrew rotational manner. What if you have the organism with having the capability of many different shapes, or no real shape at all, kind of like blobs? These are called pleomorphic. And notice the word morph is there, meaning shape. What about arrangements? Well, you've heard of no man is an island. Well, not necessarily do you find bacteria cells all alone on their own. Actually, unicellular may not always mean single cell arrangements. Each individual organism is a cell, but they may cluster together in certain patterns. These arrangements occur as a result of cell division patterns. So if I say something's diplococcal, it is cocci division to form pairs, di meaning two. Streptococci, these are cocci that form chains. Okay, so one cell after another after another. Diplobacilli is bacillus that are paired. Streptobacilli, that's bacilli that forms chains. Staphcoc are cocci that are basically round spherical structures again, that form bunches like grape clusters. That's why you get such terms as staphylococcus. And then you have palisade. And palisade is a row or a wall of bacillus. Here is the single shape, single cell, round spherical coccus. If I have diplococcus, I have them pairing up 2 to 2. Strepto, they're in chains. Staph, they're in kind of this cluster or bunch. If I have bacillus, it's single. If I have bacillus shaped with diplo, they have 2 here. They basically divided and form 2 separate cells, but they are still attached together. Strep, as you can see, is a continuous chain, and the palisades are basically as if they form a wall. And this is basically where the division occurs. Now, there are other shapes, as I've shown you, vibro, coccobacilli, spiral. A spirochete has, notice, a much tighter turns. So you get many more turns over a single cell as opposed to spiral. The stella, sort of a star-shaped structure. Now, this is an individual cell, mind you. Filamentous is kind of like, it looks a little bit like a worm, you know, and you kind of see it wiggle around a little bit maybe. And then we have pleomorphic, which, as I said, they look kind of like a blob. Now, what you see here is a scanning electron micrograph of Borrelia burgdorferi. Let me explain to you what SEM stands for, scanning electron micrograph. What this means is that this is a form of electron microscopy where it has its beam bounce off the surface so that you can get an image of a small object, and you can see the outer surface. So here we have the outer surface. Instead of looking deep inside the bacteria cell, we're looking on the surface to see that it is really a spiral-shaped structure. Borrelia burgdorferi is the infectious agent that causes Lyme disease. Now you have this pleomorphic here. This is Helicobacter pylori, and you kind of see them. They're kind of a little squiggled around, but notice that some of these also fail to completely keep a consistent bacillococci shape. In other words, they look like little sausages. But notice that they also have these extensions here, and these would be the flagella or pili. By the way, Helicobacter pylori is the infectious agent that causes gastritis and, in some cases, gastric ulcers. It can withstand the extremely low acidity inside the layer of the stomach because it sits on the surface. It causes the loss of some of the mucus that protects the stomach lining. And part of its protective mechanism is taking urea and just releasing as an exchange by its enzymes ammonium ions, which neutralize around the surface of the bacteria any of the hydrochloric acid from the stomach acid. Now we have to talk about also, how do cells divide? Well, the best known approach, really one of the only approaches for prokaryotes, there are a few others, but they're very, very remote, is binary fission. This is a form of asexual reproduction where the cell copies the chromosome and the cell basically divides. So the copied chromosome, one of the chromosome copies, I should say, goes to one cell and the other one goes to the other cell, and then you just basically pinch off the cell now into two daughter cells. Each cell is the offspring and is genetically identical to the parent. That's why we say asexual. There's no real change in the chromosomes. The process occurs in 5 steps: DNA replication, cell elongation, septum formation. Now the septum is the spacing or the separation to now pinch off the two daughter cells. The cell is partitioned and the daughter cells separate. The frequency of binary division varies from species to species, and the process is affected by nutrient availability. So when you have a very low remaining amount of nutrients, many of these cells will be basically starved, that they cannot fulfill binary fission. By the way, just to give you a little math here. Oh yes, I know some people get nervous about math, but just think about this for a second. Binary division is where you take the cell, divide it into 2. Okay? So E. coli, Escherichia coli, basically will divide every 20 minutes. If you take and start with one E. coli cell, and each hour you can basically get 3 divisions. So you start off with 3 divisions per hour, and over the period of 24 hours, you've had basically 72 divisions. So how many cells are there when you started with one cell? Interestingly enough, you will have 2 to the 72nd power minus 1, which is roughly well over 1 billion cells in just 24 hours. That's why it's important at times to be mindful of just the need for sterilization, sterile technique, and all the other things that we're going to eventually talk about down the road. Because some bacteria will rapidly reproduce even if you have only one cell left behind. Oh, and by the way, here's another example that's a little bit harder, or I should say easier to digest, but harder to imagine, is that some cells, such as tuberculosis bacteria, divide only once in 24 hours. They're slow to undergo binary fission. Now, here you see the steps. You have the prokaryotic chromosome, you have the double-stranded DNA, okay? Here's the cell. We start going through the reproduction binary fission process by first replicating that chromosome. Now we have 2 copies. The cell will begin to elongate, stretch out, add on more cell membrane, and where necessary, cell wall. You have the sequestering of each of the chromosomes, one to one side, one to the other. Now you start forming the septum, which is a wall that starts to form, and eventually it leads to the cell being completely partitioned into 2 separate daughter cells. Once these detach from each other, they are defined as separate daughter cells. Okay? Now, we have to give some consideration to the plasma membrane and the cell wall. Why? Because they function as barriers. All structures outside of a plasma membrane are considered extracellular structures, even if they're almost like virtually right intimate, attached to or connected to the plasma membrane. All structures inside of the plasma membrane are considered intracellular membranes. With the cell wall, you have the outside of the plasma membrane, and it's necessary for most prokaryotes. Why? You see, the cell wall is targets for antibiotics, disinfectants, and detergents. Penicillin alone stops the normal process of forming a cohesive structural cell wall. And eventually what happens is osmotic differences cause the cell to literally explode because more water is on the outside, rushes in, fills up the cell, fills up like a balloon and bursts. The plasma membrane, well, that's made up, oh, wait a minute. We got to define whether we're talking about bacteria or archaea. Let's start with bacteria. In bacteria, what you're going to find in the plasma membrane are phospholipids. They are developed by an ester bond from glycerol to the fatty acids. With archaea, it's a little different. They have phospholipids developed by an ether bond. This is the special bond between the glycerol and the fatty acids. So it's a slightly different approach. In the plasma membrane also, bacteria have what is referred to as linear, think of linear as straight, fatty acids. Archaea are long branched lipids. They're basically referred to as isoprene units. Bacteria can only be in a bilayer format. Archaea, though, can be either as a bilayer of phospholipids or as a monolayer format. The monolayer is due to the long lipids that are capped on either end with polar head groups. This really helps allow archaea to survive in harsh environments. Now, I have an image of this in a second, but I want you to keep in mind the differences between archaea and bacteria. Hint, hint, that's very important. Let's take a look at this. Now, when we talk about bacteria, they're only in bilayers, okay? So basically, you're going to have the phosphate groups facing outward, the watery exterior, or the watery interior of the cell. You have these nice linear fatty acids. You have an ester linkage right here that connects the fatty acids to the glycerol, which then is attached also to the phosphate group. In archaea, you have a phosphate group. You have it attached also to a glycerol, but the linkage is different. It's an ether linkage, carbon, oxygen, carbon. And what happens as a result also is that the lipids, the fatty acids, are very different. They're long branched lipids. Here's a branching here, here's a branching here, here's a branching here. And they're referred to as isoprene units. This allows the archaea membranes to be either bilayer or a monolayer. So there would be the phospholipids down here, the phosphates down here and the phosphates up here. Okay? Now, let's talk about what's on the outside of the cell, what's on the inside of the cell, and why is this phospholipid layer so important? Remember that phospholipids are what are called amphipathic molecules. The long chains here are going to exist to create what you would call a nonpolar layer. Okay? This is the lipid layer. Anything that's lipid soluble can pass through from one side to the other, but a lot of other molecules cannot and require channel proteins or some other type of passage that allows them to go from the outside to the in of the cell or the inside to the out of the cell. Interestingly enough, we have some proteins that are embedded into the cell membrane. And off of these proteins, in some cases, they're glycoproteins because they have chains of carbohydrates attached to the protein. Now, cell walls, which are on the outside of the membrane now, provide a rigid structure and extra protection from the environment. We have what are referred to as peptidoglycans, otherwise known as murins. These are the core component of bacteria cell walls. They are the target of some antibiotics which interfere in cell wall construction. As I mentioned to you before, for example, some of the importance of penicillin. Now, a pseudopeptidoglycan, pseudomurin, this type is found in the cell wall of archaea, not bacteria. Bacteria's got just murin. Pseudomurin is in the cell walls of archaea. The cell wall is very important when we talk about gram stain and acid fast properties. Both the gram stain and acid fast are means to stain to help identify what type of bacteria cell you're looking at. Okay, so it is key to telling us not only, okay, what type of bacteria it is, but in essence, what type of clinical treatment will be necessary for these bacterial pathogens. Okay, now, gram stain. This is a staining process by Gram to identify bacterial cell walls. Well, that sounds kind of makes sense. Gram was doing it, etc. Yes, but listen how it works out. Whenever you see those purple dots or those pink dots that are bacteria, you have to understand, what is this telling you? Well, gram-positive cells basically are cells that appear purple. They have a thick peptidoglycan cell wall. They have the presence of teichoic acid in the cell wall. Gram-negative stained cells appear red or pinkish. They have an outer membrane, a thinner peptidoglycan cell wall, have what is referred to as a periplasmic space or a gap between the outer membrane and the peptidoglycan layer, and have lipopolysaccharides, LPSs, in the outer membrane. Now, LPS is extremely important to understand, so make sure you're clear on this, because lipopolysaccharides contain lipid A, which is exceedingly toxic, even in extremely minute amounts, to animal cells. Okay? Gram-negative cells are actually harder to kill with chemical agents. They're resistant to lysozyme. Oh, by the way, that's the enzyme that's in tears that basically, and you can find it in other things like in egg white, etc. And it's very powerful in breaking down cell membranes of bacteria. Also, gram-negative cells have porins. These are protein channels with a pore, and this helps them to prevent entry of large molecules that could cause damage to the cells. Gram-positive cells have a thick peptidoglycan layer to retain moisture and protect the cells from crushing and abrasive forces. Gram-positive cells also have teichoic acids to bind to large proteins in the skin. Archaea can be gram-stained, but due to its flexibility of its cell wall structures, even during the transient environmental conditions, staining is not really a useful classification tool. So to help you, gram-negative, gram-positive is great to give you some basic information about the identity of bacteria cells, but not archaea. Here we can see the gram-negative and the gram-positive. Look, gram-positive, real straightforward. Big, thick cell wall made of peptidoglycans. Also, there are these unique molecules here called teichoic acid, right here, here, and here. Now, underneath them, there's going to be a small very small periplasmic space. Here you have the plasma membrane with its embedded intrinsic proteins that span across the entire from the outside to the inside of that cell membrane. Now, look at gram-negative. Very different. It's like you've got two membrane layers. 1, 2. In the middle, you have far less peptidoglycan, and you also have a periplasmic space right between the outer membrane and the peptidoglycan. You also have, instead of teichoic acid, you have these lipopolysaccharides hanging out here. You have these porin molecules. These will allow molecules to pass through and get down into here, and eventually they can pass through and get into the cell. Everybody with me so far? Remember, gram-negative is red or pink. Gram-positive is purple. Now, you can see this clear contrast here. As a matter of fact, I encourage you to take this chart and review it to be really clear about it prior to exam. Okay? And you can see what's essential and what materials are present in gram-positive versus gram-negative. Okay? When we talk about acid-fast technique, this is a detection method really that is dependent on the presence of a waxy lipid called mycolic acid. It's found in Mycobacterium and Nocardia bacteria. Mycobacterium is a bunch of different types. The best known is Mycobacterium tuberculi, the tuberculosis bacteria. Acid-fast cells stain weakly, very, very barely in gram stain. The waxy mycolic acid holds stain even in the presence of acid and will appear red or pinkish. Due to the waxy outer layer of acid-fast cells, nutrients, gases, etc. have a slow transport, and this accounts for the slow growth rates of these bacteria. Mycoplasma bacteria are small, lack a cell wall, have an enriched sterile enriched plasma membrane, and this type of bacteria normally lives inside of host cells, such as macrophages, etc. Now, there are L forms. These are reserved for bacteria that have had a cell wall but lost it somehow. Here's my mycolic acid here. Here's the peptidoglycan layer here. Here is the plasma membrane. And here is Mycobacterium tuberculosis using an acid-fast stain. So you had the stain in there, but you had to have the presence of a certain acid to enhance the dehydration and the fixing of the stain inside the cells. Here is Mycoplasma pneumoniae. Okay, this is one type of organism that causes pneumonia. The transport of substances across cell walls and plasma membrane. When I talk about passive transport, this actually goes back to ANP1, anatomy and physiology. Remember, passive transport is the transport of molecules across a concentration gradient. What we're talking about is going from high concentration to low, and it does not use energy. No pumps, no usage of ATP required. This is different from active transport, where the transport of molecules across a concentration gradient, which is usually from low concentration to high concentration. Therefore, it requires the use of energy for transport, such as pumps, or something that would use ATP for the distribution. A concentration gradient is a generalized term. It's an equal distribution of molecules, or charges, if you want to talk about positive charges and negative charges, in an environment. And usually, this gradient can be separated by a barrier. Example, plasma membrane. Okay? Now, diffusion is one type of passive transport. Basically, you're having the movement of molecules from an area of high concentration to an area of low concentration until you have a uniform, in other words, equal in concentration throughout the entire area that it's passed through. So you have a uniform distribution of these molecules. But we have several different types of diffusion that I want you to be aware of also. Simple diffusion doesn't require energy, and movement does not require carriers or transporters or anything. It's small. A lot of times you have nonpolar, that is non-charged, molecules. Some of these are like gases or lipid-soluble molecules. They basically move across a plasma membrane. So if inside the cell is low concentration and outside the cell is high concentration, if the molecules are nonpolar, they're small, they may be able to move right through the cell membranes into the cell until you have a uniform distribution both inside and outside the cell. Facilitated diffusion does not require energy, is transport from high to low concentration, but requires integrated proteins in the lipid membrane to permit transport. Here's the problem. Without facilitated diffusion, some molecules are slightly larger, might be charged or anything, but they certainly are going to be in a higher concentration outside of the cell. So how do you get them inside? You have to have these channels or these ports that will allow large molecules or such with charges to pass in to get inside of the cell. And that's what these proteins do that are embedded into the membrane. Here is an example of a transport protein. You have a high solute concentration here, a lower solute concentration here. So obviously you're going to go from high to low, and the molecules will follow in this direction. Now, with simple diffusion, you don't even need a transport protein. These tiny molecules are not polar. They are not charged, and they're small enough to wiggle between the phospholipid heads through the cell membrane. Okay? Now, osmosis. This is very important. Osmosis is the transport, a passive transport, that is, of water across the cell wall and the plasma membrane. This is the net movement, which does not require energy and is dynamic, constantly going on. That's what we mean by dynamic. As a transport always occurs even at equilibrium. So in other words, at equilibrium, you can still have molecules go in and molecules go out. But at equilibrium, they're balanced out at a certain amount going in and going out, such that inside of the cell and outside of the cell, there's no net change in the concentration. Keep in mind that any solution consists of a solute and a solvent. The solvent will dissolve the solute. Let's take a simple example of table salt and water. Water would be the solvent. Table salt would be the solute. Now, as we move through this concept, we go into the concept of tonicity. This is the amount of solutes comparing solutions separated by a membrane, such as a cell, the cell's membrane. Tonicity is really saying, okay, here is this amount of solutes versus this amount of solutes. Now, just let me help you here if you're getting a little bit lost. The amount of solutes, if it gets higher and higher, you have less solvent in the same volume of the solution. Let me repeat it again. When you have an increase in solutes in a solution, you really are also at the same time diluting down or reducing the amount of water molecules, in this case the solvent, in that solution. Now, when we talk about tonicity, we add a prefix to it to give you an idea about when you're comparing two different solutions or solutions separated by a membrane. With hypertonic, this is a solution that has a greater amount of solutes, less amount of water, compared to, let's say, the cell, what's inside that cell. Hypotonic is a solution that has a lesser amount of solutes and a greater amount of water compared to the cell. So usually when I have a cell, I put it into a certain solution. I then have to say to myself, well, what's the concentration of water and solutes that the cell is now bathing in? Now, in isotonic, iso means same. This is where you have a solution that has the same amount of solutes and the same amount of water as the cell. Therefore, there's no net movement of water. Water will always move in the direction of less water. So either inside or outside of the cell compared to the outside of the environment. So to help you to think about it this way, cell has a separation from the water. It's called the plasma membrane. Now, if there is more water inside the cell than what's outside of the cell, the water will leave the cell. The cell will begin to crinkle down as it loses some of its volume, as the water migrates out into the surrounding fluid. But what happens more often than not with bacteria is the surrounding solution is hypotonic to the solution concentration inside of the cell. In other words, there's a lot less water and a lot more solutes inside of the cell of a bacteria than outside of it. As a result, more water attempts to rush inside the bacteria cell. And when it does, it creates a certain amount of pressure. Now, if that pressure is great enough, it can cause the cell to burst. There's another term I want you to get familiarized with. It's called plasmolysis. This is the cell's plasma membrane pulls away from the cell wall due to a rapid decrease in volume. Plasmolysis, what happens, interestingly enough, is that if you have also the cell wall damage, then hypotonic solutions may cause the water to rush inside the cell. And because there is no cell wall that is holding on and controlling the amount of volume that that cell's taking, the cell begins to expand and expand and expand, and will eventually rupture the plasma membrane, and you have the lysis of the cell. By the way, gee, didn't I talk about this a little bit ago about, extremophiles or organisms that can live in extreme environments? Here's a certain group. They're called halophiles. In this case, halo for salt. These are salt-loving bacteria. They thrive in highly salty environments. They can control the amount of water that comes in, and they can also control the amount of water inside their cells so it doesn't all leave and go to the surrounding extremely salty environment, because a very high salty environment means lots of salt, not as much water. I hope that that helps. Here you can see this example here. If you have a hypertonic environment here compared to the cell inside, there's more water here than there is out here, and so the water leaves the cell. As a result, you start having the cell membrane crumple up as the volume decreases. You still have that cell wall holding up the integrity of the cell. If you have a hypotonic environment, where you've got lots of water out here, not as much water in here, so there's more solutes inside the cell versus outside the cell, water will attempt to rush in without the protective containment barrier that we call the cell wall. The cell will begin to swell, and eventually it will lyse. It will break if the cell wall is damaged. Isotonic environment, there's no net water loss or gain. The cell does not experience any real effect. It's balanced out or controlled so that the concentration of water and solutes here is the same as the concentration of water and solutes there. Now we've moved from passive transport to active transport. Active transport requires energy to pump substances against a concentration gradient. Note the word against. It is going from areas of low concentration to areas of high concentration. So there are different classes of active transport. Primary active transport, you have a pump protein. Now a pump, as you know, requires energy. In this case, the pump protein requires ATP molecules for energy to transport molecules against the concentration gradient. The secondary active transport, this is a type that relies on an ion gradient of another molecule to drive the transport. So the transport is linked to the transportation of ions from high to low. It's also referred to as a coupled transport or a co-transport. This can be in the same direction. In other words, one molecule A and one molecule B going the same direction, either inside the cell or outside. Or you can do the opposite, which is called antiport. So one goes in, one goes out. Then we also have something called the phosphotransferase transport system. This is a system that transfers high energy phosphate from one molecule, not the ATP, to a molecule being transported. Phosphotransferase systems are commonly used to import sugars into the cell and shuttle them to metabolic pathways. Here we have the example of primary active transport, and you can see that basically you've got sodium going in one direction, potassium going in the other direction. Potassium is low on the outside of the cell but high on the inside. You're delivering it inside the cell where the concentration is already high. Sodium is high on the outside of the cell, but you're going to pump sodium, which is normally inside the cell, very low. You want to pump it out with the rest of the sodium ions. Secondary transport, secondary active transport. In this case, what you have is a symport situation. Sodium, you're always transporting from high transport to low. But what gets to hop a ride on this transporter is the transport of glucose, which would be going from a concentration lower outside and higher on the inside. Finally, the phosphotransferase system, I want to get it down right. You have glucose molecules that come on inside, but once they get inside, they get phosphorylated. And you have a source of phosphorus, usually PEP, or which turns it into pyruvate. But notice it's not ATP. But what happens is as soon as you change this molecule from just merely glucose to glucose 6-phosphate, you change the substance and thereby you change the concentration gradient. The concentration gradient here will be higher. And as a result, you've just basically hijacked the system to transport glucose molecules inside. Now, we've gone from the inside for a bit. We've got to talk about the external structures. And these are structures that are involved with adhesion, movement, and protection. The flagella, singular is flagellum, is a select bacteria for some bacteria. They have a flagella for motility. So it does a whip-like movement that helps propel the bacteria into a certain direction, sometimes in response to increases in nutrients or chemicals or light or whatever is the stimulus. The flagella is a filament-like extracellular structure built primarily of the protein flagellin. Need to know that. Functions as a rotary propeller, though. It spins on a rod and ring structure, which is referred to as also basal body. And now this structure is embedded into the bacteria cell wall. For gram-positive bacteria, they have 2 rings to secure the flagellum. For gram-negative, they have 4 rings to secure the flagellum. Here's gram-positive and those 2 rings. This is in the plasma membrane and then into peptidoglycan. And here is the flagellin. And the flagellin, IN, is the single globule proteins that make up this much larger flagellum. In gram-negative, you have 1, 2, 3, 4 rings that are part and parcel for the flagellum. Now, as a result of studying the flagellum, you get into this concept called run and tumble. This is a system that allows for bacteria cells to sense and search in the environment. When we talk about something as a taxis, this is a suffix that is really indicating a response to stimuli. For example, a chemotaxis is a movement response to chemical stimulus. Phototaxis is a movement response to light stimulus. But what if it doesn't like light? That's where we refer to this as a negative phototaxis. But if it liked, or if the cell needed light, it would be responding and moving toward the light. It would be having a positive phototaxis, okay? There's also one called aerotaxis, which is a movement response to oxygen levels. Here it is running. So in other words, all of these flagella are whipping in a coordinated fashion to propel the cell. It stops at a certain point. All of these flagella are whipping around, so the cell literally sort of rotates in a position for a brief second or two. Then they realign themselves to propel themselves forward. This is how, in this zigzag approach, cells will zero in on a particular stimulus, whether it's chemical or light or whatever. Flagella arrangement is also equally important. The length and arrangement will vary by species. Some organisms are monotrichous, and that means that they have a single flagella. Lophotrichous is a tuft or cluster of flagella around one pole. Amphitrichous is one or more flagella around both poles. And then paratrichous, para is the prefix that refers to around something. This is where the flagella are distributed over the entire cell surface. As you can see here, this would be one flagella. This would be a lophotrichous flagella. This, of course, is monotrichous. Now you notice that we have the flagella on both ends. So this is an amphitrichous. And then here we have paratrichous, where it's sort of, they're around the body. Now, periplasmic flagella are located between the plasma membrane and the cell wall. Now, to help you understand this, think about it this way. They're between the plasma and the cell wall, so what do? They don't flip outside, but they cause the entire cell to rotate, to move. In this case, these flagella will allow spirochetes, remember they're kind of corkscrew shaped, to move in a corkscrew manner. So you see here is the periplastic flagellum. It kind of rotates around. And so as this is rotating, it is causing the entire cell, because of its spiral shape, to rotate or sort of spin around in a particular fashion, okay? Don't know if we're going to get this. I've been trying to work on getting some of these images where they will run. And what I'm hoping to do is get them to move around. There we are. And you can see how this is kind of wiggling around. So this is a combination, even though it is somewhat spirochete. And you can see how it is wiggling around, back and forth, okay? So let me move forward, back to the show. And that was a spirochete motility. Now, fimbriae, these are short, they're bristle-like structures that extrude from the cell surface. Think of them as sort of shorter arms, if you want to, or like rowing type of oars. I was trying to find the word, sorry about that, that stick out along the side of the body. They extrude themselves from the cell surface. They tend to be more numerous and made of protein, okay? They have adhesive properties, and they help prokaryotes stick to surfaces, to attach to each other for biofilm formation, and to attach to the host cells. They're common in gram-negative bacteria, and they're found in some gram-positive bacteria in an ARCHAEA. Examples include Escherichia coli, E. coli, SALMONELLA species that use fimbriae, to establish infections in the urinary tract, bladder, and kidney infections. See, as you can see here, these are the fimbriae here for this particular bacteria, kind of sticking out almost like spikes. And then you have a larger extension that are the pili, as you can see on this particular type of bacteria. Now, glycocalyx sugar coat. This is a carbohydrate-enriched layer. It varies among species. It allows attachment to host cells. A lot of these extensions, or the coating, the capsule coating, seem to enhance the survival of the bacteria because they're able to attach to select tissues or organs. So you want to keep that in mind. Now, the glycocalyx can be loosely associated to the cell wall. This is what you get, what we call a slime layer that helps form a biofilm. They can also be well organized, tightly associated with cell wall and forming a capsule around the cell. Now, the capsule is important because it will also prevent attacks from the immune system. The presence of the capsule increases pathogenicity, the ability to cause disease, as it promotes adhesion to the host tissues. It provides protection against host immune cells by interfering with phagocytosis. So in other words, certain cells can't just engulf them and bring them inside. Streptococcus pneumoniae, which is a leading cause of bacterial pneumonia, is 100,000 times more virulent with the capsule than without the capsule. That's how important this can be. Here you see the slime layer. As you can see, this glycocalyx, here are the cells, and they're kind of slimed out or in a sort of disorganized manner. But if you take a look at the capsule here, it's much more organized in sort of a round and uniform taking up the three-dimensional space around the cells, even when the cells begin dividing, as you can see here. Now, we've gone from the outside. We've also got to be noting some of the intracellular structures of prokaryotes. First one, cytoplasm. This is kind of the watering environment inside of the bacteria's cells. It contains like enzymes, water ions, dissolved gases, amino acids, some waste, yes, but that's going to be eventually expelled. Then we have this other unique structure called the nucleoid. It's a single circular chromosome of DNA. It's somewhat centralized, but there is no membrane that encapsulates or protects it. Also, this region of the nucleoid may also contain some RNA and proteins. Now, also what you would find floating around in the cytoplasm are ribosomes. These are organelles made up of protein and RNA. Remember that there is a type of ribosomal RNA that acts like the scaffolding that makes up the ribosome, or the ribosome subunit. And that is where the proteins will attach themselves and kind of fill out the structure of the RNA so that it can do all the necessary biosynthesis of taking amino acids and reading off the messenger RNA to form proteins. Ribosomes consist of large and small subunits, and we have means to differentiate them by the mass and sedimentation rate. Now, this is interesting. How we're able to completely understand the differences between eukaryotes and prokaryotes? In part, the sedimentation rates, and that's what is designated by that S, for both the large and the small subunit. The sedimentation rate is measured in what is called a Svendenberg unit. It's denoted by a capitalized S. So with ribosomes, it's interesting because the ribosomes in bacteria have a large subunit of about 50S. The small subunit is only 30S. Now you would say, oh, 50 plus 30 is 80. It doesn't work like that. When you have both of them together, they will have a complete ribosomal unit that has a sedimentation rate of 70. But here's the big point. If you look at eukaryotes, they differ greatly from prokaryotes. Their large subunit is 60S, the small one is 40S. Again, don't do this. 60 plus 40 is 100S, right? Nope. Together, they form a sedimentation rate of 80S. But this is for eukaryotic ribosomes. Now, why is this so important? Because ribosomes are the key target for bacterial antibiotics. That's one of the key structures, one of the key targets for antibiotics. When we get into the antibiotics chapter later on in the course, you're going to see that. Okay? Now, we also have, interestingly enough, a cytoskeleton. It provides prokaryotes with sort of structure and support. It helps organize binary fission, and it provides a general organization of cytoplasm for biochemical processes, movement of enzymes and other things throughout the cell. Now, the bacterial cytoplasmic elements include homologs to the three major types of eukaryotic cytoskeletal proteins, and that is actin, tubulin, and intermediate filament proteins. There's also a fourth group, the MIND-PARA group, that appears to be unique only to the bacteria. Okay, so remember all of this that we talked about here? We have the FIMBRIAE, the FLAGELLA. You might have an inclusion body here, a ribosome. You will have, of course, the nucleoid region, etc. You will have the plasma membrane, the cell wall. Okay, these are all very important. Here we have also a look at not only the nucleoid region, but you're going to see also the ribosomes. 50S, 30S, complete 70S, that is a bacterial ribosome. What about inclusion bodies? Can we get a little bit more about that? Sure. See, inclusion bodies, remember, they don't alter a solution's solute concentration. They're a distinct collection of substances that exist inside of prokaryotic cells. Now, some of them are going to be insoluble granules, and sometimes they're bound inside of a membrane. The function is really storage sites, and so you get glycogen, polybeta hydroxybutyrate, which is another form of a carbohydrate, okay? Volutin, otherwise known as metachromatic granules. These are interesting, and they do offer some unique coloring when you look at the bacteria. They contain polymerized phosphate. Cyanobacteria use protein-coated inclusions called carboxyosomes. They're loaded with carbon-fixing enzymes, especially rubisco. If you're not familiar with rubisco, this is one of the key enzymes that plays a massive important role in photosynthesis. Then we have also magnetosomes. These are magnetic iron-rich inclusions. They're encased by a membrane, and they may be used to protect against oxidative stress from toxic forms of oxygen. One other thing to keep in mind, this may be theoretically, and there's been some speculation, some research in this area, where magnetosomes may help sort of provide an internal magnetic compass for some types of bacteria. Okay, here's some examples of the inclusions. Here, 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 here. Okay, here are some of the carboxysomes that you see here. And here are the magnetosomes, as you can see those little dots there. Okay, now we are finishing up on the prokaryotes, but we've got to talk about one other thing. Prokaryotes, some of them, have a very unique capability to withstand extremes by basically shutting down. It's almost like the sci-fi movies where they talk about, well, someone was in suspended animation for 100 years, and they just came out of suspended animation, and this is what happened. Let's take this logically, factually, but it's very interesting because it helps us to understand how some infections can die out, but they're really not dead. They just need the right conditions to reactivate and become infections again. I'm usually talking about infectious agents, pathogens in the environment. I'm not talking about it necessarily in the person. These are called endospores, otherwise known as bacterial spores. Now, don't get them confused with spores from certain types of protozoan or spores from fungal cells. Very, very different, but we'll get into those later on. Endospores are metabolically inactive spores. They allow certain cells to enter a sort of a dormant state. An endospore that's released is called a spore. Vegetative cells are actively growing cells. Bacteria spores are highly resistant to environmental stresses. That includes starvation, heat, radiation, drying, freezing, and various chemicals. Most spore-forming bacteria are gram-positive. There are a few that exist that are gram-negative, such as Sporomusta ovate. Most medically relevant bacteria include the bacillus and the clostridium. For example, Bacillus anthracis. One of the ways that anthrax was distributed was not in a kind of liquid slurry, but they just made the bacteria go into the spore state. And that way you could store them for a period of time, disseminate them as spores. People would breathe them in and then get anthrax. Clostridium. This may be familiar with you. You may have heard of Clostridium tetani, the bacteria that causes tetanus. One can get, by working with the soil, by working outside, getting cuts and everything else, some of the spores get into some of the injuries, and then they become vegetative cells again. They become actively growing, actively producing toxins, causing disease. Clostridium botulinum, the same situation. Clostridium botulinum, very common in soil. And so that's why if you take and harvest your fruits and vegetables, if they've had soil splashed on them, you have to wash them. And in some cases, if you're going to store them for a long period of time, you use specialized canning procedures to kill off the Clostridium botulinum spores. Otherwise, people will die from the botulism toxins that are produced from the reactivated Clostridium botulinum. So, yes, this is a process that allows medically dangerous strains of bacteria to survive. Now, I just mentioned two, tetanus, botulism, but let's go over to a few other ones. And yes, you need to know these. Clostridium perfringens. This is the pathogen that causes gas gangrene. You have an infected wound, and what will happen is they start releasing certain toxins that destroy the tissue, can lead to gangrene. But not just this simple gangrene, it also erupts producing certain toxic gases, and these gases allow for basically the bursting of certain injuries because of the presence of the gas gangrene bacteria. Clostridium difficile. This is a terrible, severe diarrhea that occurs. Most commonly, you will see this when an individual has received antibiotic therapy and they're in the hospital. The antibiotic therapy causes a dysbiosis, a disruption of the bacterial microbiota. And so the disruption favors now Clostridium difficile, and the patient gets this, what's also referred to as C. diff, and suffers tremendously. And of course, it's very, very messy and very, very contagious. And you have to go through all sorts of decontamination because it's a real difficult time to clean up the mess and prevent anyone else from getting it. Now, let me introduce you to a term, sporulation. This is the process of forming an endospore. Now, when we take a look at this, we have favorable environments. The endospore germinates into a vegetative cell. But let's say the endospore has to start forming because of stress. Maybe it's extreme drying now in the environment, or the nutrients have been used up. So basically, the only way that, in essence, this species can survive is to basically form an endospore. Now, you can see endospores in certain types of bacteria also. If you look here, you have the exosporum here, which is basically the remnants of the vegetative cell. You have the spore cortex here, and then the core amount in here, which contains the DNA and some other essential materials, okay? Endospore staining, there are special stains for them, particularly malachite green. And what happens is it doesn't necessarily always completely penetrate the cell, but it basically adheres onto the spore sufficiently. so that you can make a distinction of what is a endospore versus what is a vegetative cell. Okay, now here's another endospore, and I think this one is anthrax. Either that or it's Clostridium tetani. But what you can see is part of the cell gets sequestered and saved only for what is essential. And that will include, of course, the nucleoid with the chromosome. Now, the sporulation steps. This is vegetative growth stops. I'm sorry about the barking of my dog in the background. I apologize for that. DNA is duplicated, and a septum, a wall, forms inside the cell. Now, this is going to be an asymmetrical division. It's not binary fission. So you're going to have big part of cell, a tiny part of cell. The larger part of the cell is called the mother cell, which will engulf around the smaller cell. That smaller cell is going to be eventually the endospore, but it's at this point called a forespore. Now, the forespore has a layer of peptidoglycan laid down between two membranes. Other cell makes proteins that will form this cell a spore coat. So the mother cell starts producing more of these proteins that help form the spore coat. What will also be built into this forespore coat is calcium and dipicolinic acid. I want you to keep those 2 points in mind because it's very important. Those are important components that account for the formation of the endospore. Eventually, the mother cell basically dies. It disintegrates, is degraded, and the endospore is now released. As you can see in this diagram here, the cell stops growing, the DNA is duplicated. Now, you start forming a septum here. You have DNA here, DNA here. This is going to be eventually what the endospore. And so now this larger cell is going to basically direct all of its biochemical machinery to making the proteins and making the other substances necessary for that protective coat for this forespore. And eventually what happens is you continue to have more of this built up in the forespore here. And it's going to include peptidoglycan material. It will have calcium, picolinic acid, and eventually this cell will atrophy and die. This core wall and its cortex will protect the essential genetic information on the inside, and this cell will be able to be reactivated when the environment is much more favorable. Okay? Here is Clostridium tetani. They refer to this as sort of the drumstick or tennis racket. Here is the spore that's forming here. Okay? As I said, this is a very common soil bacteria, and it's the causative agent of tetanus. When you see this growing in the soil, C. tetani is rod-shaped, and it's up to about 2.5 microns long. However, when you start forming spores, the C. tetani becomes substantially enlarged at one end. So this is the widening here, is the formation of the endospore. Okay, we've gotten through all of this, and that's great. By the way, I would encourage you, wherever you have certain essential information, to do a process I call hot paging. Either attach a little tab to that particular area, or attach a note, or maybe even write it in the inner cover of your book. Okay, this is important for information about endospores. This is the information about reproduction of protozoans. The same thing will go on when we deal with eukaryotes in the next lecture. What I do want you to do is take your medical microbiology book and review the visual summary on page 88, the chapter overview on page 89, the end of chapter questions, which are on pages 90 to 92. Now, in Mastering Microbiology, you're going to have a homework assignment this week. You will have also a quiz that covers this material. I encourage you also to peruse, that is, review some of the videos that are in the Blackboard for the lecture content. And then in our next section, we will do the introduction to eukaryotic cells, which is chapter 4. I always encourage you to pre-read the chapter before you listen to the lecture. That will help to reinforce your memory about the significant information. Well, that's it. Have a great day.